It is my great pleasure to be here today. I should introduce myself first, and then I'll introduce our speaker. So my name is Jenny Bruin. I'm an associate professor in the Department of Biology and Institute of Biochemistry at Carleton. And I run a research lab that focuses on diabetes. And I'm here today just to moderate and introduce one of the PhD candidates in my lab. So Lahari Basu is a PhD candidate who started in September of 2021 in the midst of the pandemic. So a bit of a funny first year, but she's off to the races. So Lahari did her undergraduate, her bachelor's at the University of Western Ontario, and then came to my lab here in Ottawa. And she's working on a project investigating the off-target effects of a chemotherapeutic drug called Cisplatin. But I won't tell you any more than that. I'll let Lahari take over from here and tell you about her project.
Thank you so much for that introduction, Jenny. So today I want to talk a little bit about my PhD research, which is essentially understanding if there is a potential link between cisplatin, which is a chemotherapeutic agent, and diabetes risk. Now, essentially, currently, there are over 537 million adults around the world living with diabetes, meaning that almost 1 in 10 people are impacted by this condition. Now, when it comes to diabetes, there's 2 types of diabetes: type 1, which is an autoimmune disease, and type 2, which can be developed later in life, and is also the focus of my talk today. Now, of these 530 million cases, approximately 90% of them are type 2 diabetes. But what exactly is diabetes? Well, it all starts with an organ in our body called the pancreas. Now, the pancreas hosts clusters of cells known as islets, and these islets are actually made up of different hormone-secreting endocrine cells, or hormone-secreting cells, denoted by the different colors in this diagram. Now, one of the most important cells in the islet, and the one that I'll be focusing on today, is the beta cell. Now, what makes the beta cell special is that it's able to use our blood sugar, or glucose, and use it to produce and secrete a hormone called insulin. This insulin then goes and binds to other cells in our body, such as liver cells, muscle cells, and fat cells, and it tells these cells to use up any extra blood sugar in our bloodstream and use it as energy so that these cells can carry out their regular cellular functions. In turn, with an increase of insulin being secreted by the beta cell, there's a decrease of blood sugar in our bloodstream. Now, between meals, there's relatively low glucose levels in our bloodstream. However, because there's still some glucose, we still see some insulin secretion. But right after a meal, there's an increase of blood sugar in our bloodstream, prompting the beta cell to produce more insulin so that this blood sugar can be used up by other cells in our body. Now, taking a closer look at the inner workings of the beta cell, essentially how this works is that the beta cell takes up glucose from the bloodstream, and through a series of reactions powered by the mitochondria, it actually uses this glucose to produce energy. This energy then goes and triggers several chemical reactions, which ultimately leads to the secretion of insulin. Now, when glucose is increased in the bloodstream, these pathways are highly activated, causing more insulin to be secreted. Now, sometimes what can happen is that the beta cell is either unable to sense glucose or it's unable to produce a sufficient amount of insulin. This in turn leads to a couple of things. First and foremost, it leads to a rise in our blood sugar levels. Now, because the blood sugar is essentially the trigger for the beta cell to try to produce insulin, elevated levels of blood sugar prompts the beta cell to try to ramp up insulin production. But because this beta cell is experiencing dysfunction, it's unable to do so. And the blood sugar levels remain elevated, and the beta cell tires itself out trying to produce enough insulin to lower these levels. This in turn leads to an overworked beta cell, which causes blood sugar levels to remain elevated and is essentially type 2 diabetes. Now, type 2 diabetes is more than just elevated levels of blood sugar. Type 2 diabetes can lead to several adverse health outcomes, including stroke, cardiovascular disease, kidney disease, and can even prove to be fatal. Now, I mentioned 1 in 10 people around the world are experiencing diabetes, and this is quite a large subset of the population. But there is a unique subset of the global population that's at an even higher risk of developing type 2 diabetes, and these might be cancer patients. Now, a study done in 2018 found that cancer survivors actually had an increased incidence of type 2 diabetes compared to individuals that had never been diagnosed with cancer over the course of 10 years. Another study looked at the different causes of mortality in cancer survivors after they'd enter remission, and compared to causes of mortality including cardiovascular disease, respiratory disease, and kidney disease, cancer survivors that had diabetes had almost a 17 times higher rate of mortality than any other cause of death. Now, chemotherapy is one of the most commonly used treatment options for cancer patients. And essentially how it works is that anti-cancer drugs are administered to cancer patients intravenously, and these drugs then go and target cancerous cells in the body, killing them before they're able to replicate and multiply. Now, because these anti-cancer cells are administered intravenously, they travel throughout the body. They're not localized to just the cancer cells themselves, which means that it has the ability to have off-target effects or effects on cells that aren't necessarily cancerous. Cisplatin is one of the most commonly used chemotherapeutic agents used today, and for good reason. It's extremely effective at killing cancerous cells fast. It's used to treat a wide variety of cancers, including testicular, breast, lung, and colorectal cancer. Now, one of the key features of cisplatin is that it has a platinum core, which allows it to be so effective at killing cancerous cells. But because it's based with this, it has a core of this heavy metal, it can have several severe off-target and toxic effects to our body. A study done in 2019 looked at the platinum levels in cancer survivors' bloodstreams after they had received cisplatin treatment. Now, each one of these dots on this graph is actually the measurement of platinum levels in a cancer survivor's bloodstream. And what we can see from this graph is that almost 25 to 30 years after the end of their treatment, there were still highly detectable levels of platinum in their bloodstream. Now, having a heavy metal present in your bloodstream for this long of a period of time can be extremely toxic to the body and can have negative impacts on our cellular functions and metabolism. Other studies done throughout the years have shown that there is a link between cisplatin exposure and reduced metabolism and development of metabolic syndrome, which is a known precursor to type 2 diabetes. But this link between cisplatin exposure and how it might impact diabetes risk in cancer survivors and how it might impact beta cell function is not well studied at all, which prompted my PhD research, which is essentially understanding if cisplatin impairs beta cell function. And if it does, how exactly is it doing this? What are the underlying mechanisms by which this is occurring? Now, to first study this question, we used a tool called Immortalized cell lines. And essentially, immortalized cells are cells that are grown within a dish, and they're very commonly used in toxicology research. Now, these immortalized cells are quite popular because, one, they have an unlimited lifespan, and two, because they're almost identical to one another, they provide very reproducible results. However, one of the biggest drawbacks of these immortalized cell lines is that because they're grown in a dish, they're not exactly like the cells that we see in our body. However, they provide a great foundation for trying to understand this question of if cisplatin is impairing beta cell function. Now, to try to answer this question, we first used 2 cell lines, a colon cancer cell line that was donated to us by the Mackay Lab here at Carleton. And these colon cancer cells were actually known to be cisplatin sensitive. So we knew for sure that these cells would die when they were exposed to cisplatin. We then compared the activity of these colon cancer cell lines to the beta cell line and exposed both of these cell lines to low, moderate, and high doses of cisplatin or no cisplatin for 48 hours to see if cisplatin impaired beta cell function. Now, this research was actually conducted by an undergraduate honors thesis student in our lab in 2018, Andrea Smith. And the first thing that she looked at was cell death in response to cisplatin in these immortalized cell lines. Now, looking at this colon cancer cell line, the cell line that we know for sure is sensitive to cisplatin, a high dose of cisplatin was required to cause significant cell death compared to the control, where their cells were not exposed to any cisplatin. In contrast, in the immortalized beta cell line, when exposed to all three concentrations of cisplatin, low, moderate, and high, there was actually a significant increase in cell death compared to the control, meaning that these immortalized beta cells were actually more cisplatin sensitive than that colon cancer cell line. Now, zooming in onto the immortalized beta cell line, we next wanted to measure insulin secretion, since insulin secretion is a marker of beta cell function. Now, typically, what we would expect to see in insulin secretion is that under low glucose conditions, or essentially between meals, there's some insulin secretion, but under high glucose conditions, or right after a meal, there's even more insulin being secreted. When we expose these immortalized beta cells to low, moderate, and high doses of cisplatin, we actually see the opposite effect, where we're seeing increased insulin being secreted under low glucose conditions and decreased insulin being secreted under high glucose conditions, which is extremely odd behavior from these beta cells. Now, all of these data goes to show that cisplatin is having a profound impact on beta cell function in immortalized beta cells. But as I mentioned before, these cells are different from the beta cells that we see in our body. So we next wanted to look at the impact of cisplatin on isolated mouse islets to see how cisplatin impacts beta cell function in the context of the whole islet. Now, these islets were isolated from mice, and mouse islets are actually quite similar to human islets, which provides us a good foundation of understanding how these results might translate to humans. Another facet about using isolated mouse islets over immortalized beta cells is that the isolated mouse islets are intact. The islet isn't just the beta cell, it's a cluster of different cells. So understanding how cisplatin impacts beta cell function in the context of the entire islet is also extremely important in order to answer this question of if and how cisplatin is impacting beta cell function. Now, we isolated islets from male and female mice, and we exposed them to a moderate dose of cisplatin or no cisplatin over the course of 48 hours. We again measured insulin secretion, and as a reminder, under low glucose conditions, we expect to see some insulin being secreted, and under high glucose conditions, we expect to see an increase in insulin secretion. And what we found is that in cisplatin-exposed male mouse islets, there's increased insulin secretion under low glucose conditions, similar to what we saw in the immortalized beta cell lines. And there is some, like a slight increase in insulin secretion under high glucose conditions, but nowhere near as dramatic as what we see in the control islets. And this pattern is again repeated in female islets as well, where we see that there's an increase in insulin secretion in cisplatin-exposed islets under low glucose conditions, and a slight increase in insulin secretion under high glucose conditions, but not as dramatic as the control. Now, measuring insulin secretion this way does provide us detail on if beta cell function is being impacted by cisplatin exposure. But insulin secretion is a very dynamic process, and these data just shows insulin secretion at a single snapshot in time. So the next thing that we wanted to do is we wanted to track insulin secretion dynamically in both control and cisplatin-exposed islets. Now, this was done by a previous lab technician in our lab, Kaylee Rick, and a current PhD candidate in our lab, Miriam Hoyak. And what they did is that they measured insulin secretion every 2 minutes in mouse islets in response to low glucose, which is denoted by the white areas of the graph, high glucose, and potassium chloride. Now, what we typically expect to see is that under low glucose conditions, there's some insulin secretion, but when the mouse islets are exposed to high glucose, there's a rush of insulin being secreted, causing this peak. And while the islets are still exposed to high glucose, there's elevated levels of insulin being secreted. The islets are then exposed to low glucose once again, bringing the insulin secretion back down, and then it's exposed to potassium chloride. Now, potassium chloride is a potent stimulator of insulin secretion, and essentially it triggers the maximum secretion of insulin, causing a rise in insulin secretion much higher than that of what we see when exposed to high glucose, and causes this insulin secretion to be elevated while these islets are exposed to potassium chloride. We finish off this assay by exposing the islets to low glucose once again, causing this insulin secretion to come back down. Now, in cisplatin-exposed islets, we see something a bit different. Under low glucose conditions, cisplatin-exposed islets had relatively similar levels of insulin secretion to the control islets, but it was trending to be slightly higher. When exposed to high glucose, there's no rush of insulin being secreted, which is a key feature of beta cells. Insulin secretion is elevated throughout high glucose, but that lack of a rush of insulin being secreted is something interesting to note. When the islets were exposed to low glucose once again, cisplatin-exposed islets took some time to lower their insulin secretion. And what's very interesting is that when exposed to potassium chloride, that potent stimulator of insulin secretion, cisplatin-exposed islets seem to have almost no effect at all. Their insulin secretion stayed low at near basal levels, what we see under low glucose conditions. However, after the cells were exposed to potassium chloride and they were exposed to low glucose for the third and final time, we see a peak in insulin secretion in cisplatin-exposed islets, which may actually be a delayed effect of this potassium chloride. Now, all of the data so far has shown that cisplatin is having a profound impact on insulin secretion and beta cell function. But how exactly is it doing this? Well, coming back to this diagram, the mitochondria plays a very central role in using the glucose from our bloodstream and harnessing it to generate energy to trigger reactions to cause insulin secretion. So is it possible that cisplatin is reducing the mitochondrial function, in turn causing decreased insulin secretion? Well, we can measure mitochondrial function using an instrument called the Seahorse Analyzer, which we had access to thanks to Dr. Jan Menningen at U Ottawa. Now, how the Seahorse Analyzer works is that it measures oxygen consumption rate or mitochondrial function over time, and in response to 3 injections of 3 compounds. The machine first starts off by measuring basal mitochondrial function, what we typically expect to see from a resting cell, and then it injects the first compound. When the first compound is injected, mitochondrial function is reduced, causing oxygen consumption rates to come down. The machine then injects a second compound, which causes oxygen consumption rate to go up and promotes the maximum function of the mitochondria. A third and final compound is added, which causes the complete inhibition of the mitochondria, reducing all oxygen consumption rate down to just that of non-mitochondrial components of the cell. Now, in control mouse islets, we see a curve that we typically expect to see. Under basal conditions, there's strong mitochondrial function, and when compound 1 is added, the mitochondrial function is decreased. When compound 2 is added, mitochondrial function rises back up to maximum levels, and then when compound 3 is added, mitochondrial function is essentially diminished. In cisplatin-exposed islets, overall, what we can see is that mitochondrial function is quite reduced in these cisplatin-exposed islets. In fact, under basal conditions, the mitochondrial function of cisplatin-exposed islets were actually 4 times lower than that of the control islets. In response to compound 1, we see that in cisplatin-exposed islets, there's a slight decrease in mitochondrial function, but nowhere near as dramatic as what we see in the control islets. When compound 2 is added, we do see a rise, but again, nowhere near as dramatic as what we see in compound 2, in the control islets. So overall, what we see is that cisplatin is impacting mitochondrial function. And this in turn may actually be why we're seeing such dysregulated insulin secretion in cisplatin-exposed islets. Now, when we studied these isolated mouse islets, we're studying the direct impact of cisplatin on these mouse islets. But within our body, the islet isn't isolated. Everything in our body is connected, which is why we wanted to study the impact of cisplatin in an in vivo mouse study. in vivo just meaning in the body. And using a mouse study allows us to study the impact of cisplatin in the context of the whole body, because at the end of the day, all of our organs and our cellular functions are interconnected. And it's important to understand how cisplatin would impact beta cell function in the context of everything together. Using a mouse study also allows us to assess beta cell function over longer periods of time, rather than just the 48 hours we've used for the immortalized cell lines and the mouse islets. Now, for our mouse study, what we did is that we exposed male and female mice to cisplatin or saline every other day over the course of 2 weeks for a total of 7 injections. Now, why we used this treatment protocol is because this is similar to what a human cancer patient would receive, where they receive multiple small doses of cisplatin over the course of several months. Now, one thing to note is that the total dose of cisplatin used in this study was lower than the typical human dosage, simply because the lowest human dosage would be fatal to the mice. However, even with this lower dosage, we actually see some very interesting results on beta cell function as a result of the cisplatin exposure. The study was a short-term study, meaning that we only looked at beta cell function a short period of time after they'd received cisplatin, only a couple of weeks. One of the metabolic assessments that we do during a mouse study is called a glucose tolerance test, where we measure blood glucose and plasma insulin levels in response to an injection of glucose. Now, what we typically expect to see in terms of blood glucose levels is that before they receive an injection of glucose, mice blood glucose levels typically hover between 5 and 10 millimole per liter. But once they've received that injection, this blood glucose actually spikes and then comes back down over time. And this complements what we see in plasma insulin, where before mice receive an injection of glucose, they have relatively low levels of plasma insulin, but after they receive that injection of glucose, there's a peak in plasma insulin levels corresponding to this peak in blood glucose, because it's trying to lower the blood glucose levels. And as blood glucose levels come down, plasma insulin also comes down as well. Now, when we did this in cisplatin-exposed mice, in male mice, we don't really see a difference in blood glucose levels between the control and cisplatin-exposed mice. Their blood glucose levels seem to hover very close to one another. In female mice, however, 15 minutes after that injection of glucose, cisplatin-exposed females actually had significantly lower blood glucose levels than that of control females. The plasma insulin data is actually even more interesting. In cisplatin-exposed male mice, we see that 15 minutes after they've received that that glucose injection, their plasma insulin was actually significantly reduced, even though we typically expect to see their plasma insulin levels peak at the 15-minute mark. And this is repeated in female mice as well, where we see a significant reduction of plasma insulin at the 15-minute mark. So these data goes to show that cisplatin is having a profound impact on beta cell function, both in vivo or in the body in mice, as well as outside of the body directly on the mouse islets. We also know that cisplatin is impairing beta cell function in mice in a non-sex specific manner in both males and females equally, and it's also reducing mitochondrial function of these beta cells, which may be contributing to why we're seeing such dysregulated insulin secretion. But what still needs to be answered? Coming back to this graph, what's very interesting here is that this lack of response to potassium chloride in cisplatin-exposed islets, that potent stimulator of insulin secretion, isn't stimulating any insulin secretion at all in these cisplatin-exposed islets. Now, one reason this might happen is because of dysregulated electrical activity. Although the mitochondria plays a central role in using this glucose to trigger insulin secretion, the electrical activity of beta cells also plays a role in this, where certain levels of electrical activity are needed for the rest of these reactions to occur in order for a sufficient amount of insulin to be secreted. Now, we can measure electrical activity of beta cells using something called electrophysiology, where an islet, or just a beta cell, is hooked up to 2 probes, and we can measure the electrical activity of the cell in response to low glucose, high glucose, and potassium chloride. And this is actually something that I'm planning on setting up with Dr. Melissa Chi here at Carleton in the future. Another player of why we might be seeing cisplatin-induced beta cell dysfunction is DNA damage. Just with any cell in our body, the beta cell also has DNA, and this DNA essentially tells the cell what it is and what it has to do. If cisplatin causes damage to this DNA, it's possible that the beta cell can experience dysfunction and also experience cell death, causing that reduction in beta cell function. Now, we can measure DNA damage through something called histology, where we'd harvest whole pancreas from mice exposed to cisplatin or saline, embed tissue in the paraffin wax, section these tissue and stain them with fluorescent antibodies, and then image these sections. And what we get are images like this, where each of these circles are essentially an islet, and the different colors denote if beta cells are expressing high levels of DNA damage markers. And this way we can visually see if cisplatin is impacting beta cell function by increasing DNA damage. And we're also able to quantify the percentage of beta cells that are experiencing this DNA damage. Now, my mouse study that I've done so far looked at the short-term impact of cisplatin. But what about the long-term impact of cisplatin? I plan on repeating my mouse study, but tracking the mice for a longer period of time after they've received cisplatin to see if perhaps they recover, perhaps their beta cell dysfunction recovers, or perhaps it gets worse. I also want to know what happens if we pair cisplatin exposure with a known stressor of type 2 diabetes, such as high-fat feeding. Now, obesity has been very closely linked with type 2 diabetes. So is it possible that if we expose obese mice to cisplatin, that the combination of high-fat diet and cisplatin actually exacerbates beta cell dysfunction and in turn increases the risk of developing type 2 diabetes? Now, mouse islets are an extremely valuable tool of understanding how cisplatin might impact beta cell function in humans. But although they're similar to human islets, they do have some key differences. So it's important to understand how exactly cisplatin impacts human islets directly. I plan on exposing human islets that have been isolated from donors in order to study if we can reproduce these results that we see in mice in the human islets. And at the Bruin Lab, we're very lucky to be able to work with the Alberta Diabetes Institute islet core, which provides us with human islets from donors, which we can use to study the impact of cisplatin on human beta cell function. Now, after we've gathered all of this data, the next step is essentially to understand how can we predict beta cell function during cisplatin treatment. We don't want to say that we're stopping chemotherapy in order to protect beta cells, but rather, what can we do to make sure that both cancerous cells are targeted, but beta cell health is still protected? We want to run a mouse study that pairs in vivo cisplatin exposure with anti-diabetes medication in order to see if that helps protect beta cell function, or potentially pair cisplatin exposure with healthier lifestyle choices, such as an improved diet or a low-fat diet, in order to see if these are measurements that we can take to protect beta cell function during chemotherapy. Now, with that, I want to thank you all so much for listening. Thank you to my lab for supporting me in my work, especially Dr. Bruin. Thank you to my collaborators for helping me generate this data. And a very big thank you to Diabetes Canada for funding my research and allowing me to continue on with this project.
All right. Thank you, Lahari. That was fantastic.
Thank you.
So we have lots of time for questions. There's one in the chat already that I can read out, but I just want to let any of the participants know if you have questions, you can type them into the Q&A. I'll read them out to Lahari and she can answer them. So we'll start with the first one that's there. So the question is, and I think you may have actually answered it with your last slide, but have you or are you intending to see if any factors can reduce the effect of cisplatin on beta cells?
Yes. So yeah, that's actually one of the goals of our study is understanding, like, after we've accumulated all of this data on how cisplatin might be impacting beta cell function, trying to pair cisplatin exposure with either anti-diabetes medication or healthier lifestyle choices to see if implementing these secondary measures with chemotherapy can help protect beta cell function. And that is something that we're hoping to do in the future.
Excellent. Okay, and there's another question here. Have you seen how diabetic ketoacidosis, or DKA, how a DKA reaction has resulted by chemo on a diabetic patient? So I think the idea there is if the person has diabetes in the first place, then what would be the impact of chemotherapy?
So I haven't specifically looked at that. However, there are a few studies that have shown that individuals with diabetes are at a higher risk of developing cancer, not necessarily because of the diabetes, but simply there's just an increased incidence of adverse health outcomes because of the diabetes, and cancer may be one of them. So in terms of that, however, there isn't a lot of research done to see if chemotherapy exacerbates an already known case of diabetes, but that is an interesting point of research for the future.
Yeah, very interesting question. Another interesting question, could CoQ10, and I think that's coenzyme Q10, which is an antioxidant, I believe, be used to improve mitochondrial function?
Potentially, that's actually something I didn't mention it in this presentation, but that is something that we're planning on looking and seeing and using that seahorse analyzer for, is seeing if we co-treat these cisplatin exposed islets with certain protective measures of mitochondrial function, if that actually improves their mitochondrial function. And so Q10 might actually play a role in sort of helping protect the mitochondrial function of these beta cells that are exposed to cisplatin.
Yeah, great questions. We'll give people a few more minutes. If anybody else has questions or comments, please feel free to put them in the Q&A. I'm not seeing anything pop up. I would also encourage participants who are interested, if a question comes to you later, please feel free to reach out to Lahari or myself. I mean, my email is certainly on the website for the biology department, and I can also put you in touch with Lahari. So we'd be happy to answer questions later if they come to you. But I think maybe we'll wrap up there. Thank you very much, Lahari. That was outstanding.
Thank you, Jenny, for helping me answer those questions.
And I guess thank you to the Faculty of Science for letting Lahari share about the research happening in our lab.
Yeah. All right. Well, thank you so much once again to everyone for listening to our talk. I hope you enjoyed it, learned something new, and yeah, always feel free to reach out to either myself or Jenny if you have any questions about this.